Our next presenter is Dr. Kenneth Berns. Dr. Berns is the director of the Genetics Institute at the University of Florida. Dr. Berns has devoted most of his scientific research career to the study of the molecular basis of replication of the human paravirus, adeno-associated virus. He has had many awards. He has made enormous contributions to the field of research, and then he interrupted his research career for a little sojourn in New York City, and he decided that he preferred to continue work at the bench and to do the work that he so loves. And so he's going to tell us today about the work that he so loves. And are you going to mention the difference between one career and the other, and how good it was to go back to research?
Possibly.
Excuse me. Let me mic you. You'll be the walk around, so I'll just mic you. So.
You want to do that?
Yeah, I mean, if you're planning to walk around, it will be easier.
Just put it on. Maybe on this, yeah, the tie is fine. Hopefully we'll get any feedback.
Yeah, I'll try that.
Okay. Well, thank you, Marianne, for your kind comments and for the invitation. First to be on the board as one of the original members, and now to give this presentation. I have no audiovisual aids, having seen the trouble people have gotten into by putting their life into the electronic media recently. I decided that I would just do this all verbally and let it go at that. The thing that I start off with is the fact that when I was growing up in Cleveland, I used to deliver newspapers. And the weather is atrocious. And I would walk around my route through about 6 inches of snow, and my toes were killing me. And I used to mutter under my breath, when I grow up, I'm going to be a physician and go live in Florida. Now, I didn't anticipate that would happen 3 times. But in fact, that is what happened. I was an undergraduate at Harvard. And after my junior year, my tutor, Jack Fresco, some of you may know, a longtime professor at Princeton now, sort of took me by the arm and said, you're going to spend the summer after your junior year in Paul Doty's laboratory, and he arranged an NSF fellowship to do that. And it was a life-changing experience from my point of view. My classmate who was with me in the lab was Bruce Alberts, and there were some other notable characters, Bob Hazelcorn, Carl Schildkraut, and others who were graduate students in the lab at that time. And I learned a good scientific lesson. And that is that we were looking at UV irradiation of DNA. And I had a project, and I finished the project in less than the 10 weeks that I had. So Julius Marmar, with whom I was working, said, you know, we have all these DNAs of different GC contents, and we've characterized how GC correlates with the melting temperature of DNA. So why don't we see what happens when we UV irradiate all these different DNAs and what effect that has on the melting temperature? And the model at that time was that the lesion caused by UV involves cytosine and the hydration across the 5-6 double bond. And so he did the experiment, expecting the high GC DNAs would have the biggest effect. And of course, we got completely the opposite result. And nobody could really explain that, but I remember sitting down with Professor Doty and Julius, and we talked things over. I came up with an alternative explanation, which I won't bore you with, which had nothing to do with reality. And so nothing happened until the next year when the Dutch discovered that the real lesion was the thymine dimer, and suddenly our data were really quite supportive of that finding. And so I got my first paper as a result of the sort of PS on the end of my summer project. And the other thing that happened at the end of my summer project was that a fellow named Charlie Thomas, who'd been at Cold Spring Harbor at the phage meeting, came through the laboratory. I met him. He had gotten his degree in Doty's lab. And I left after my junior year to go to Hopkins for an accelerated medical program. It was supposed to get me through early. So it only took me 7 years to get an MD instead of 4, the usual way of things. And that was because it turned out when I got there in the first year, I didn't have to take any courses. And so I spent the whole year working in Charlie's lab. And we actually had some very good results having to do with the structure of T4 phage DNA. And one of the things we showed was that the complementary strands of the double helix were single polynucleotide chains uninterrupted, which, considering that nobody knew how big DNA could be in those days, this was 50 years ago, was rather an interesting finding. And we published 2 papers in Journal of Molecular Biology, which were the high point of my scientific career in my mind, because they were the 2 papers that we published, and the next one was by a couple of Frenchmen named Jacob and Monod, describing something called the operon. And so we were in very good company through little fault, I might say, of our own. Anyway, I went to Hopkins and did all this work with Charlie. And so already I've mentioned a few mentors. Fresco, Marmar, Charlie Thomas. And at the end of that first year, we had signed up to go to the phage meetings, and we'd asked for 45 minutes to talk about our results, which was, those days you talked as long as you wanted to, as long as you told them ahead of time. That was great because we had a lot of things we'd done, until Charlie told me that he was going with his wife to tramp in the foothills of the Himalayas, and that I was going to give the talk. And I was just sophisticated enough to know how much trouble I was in at that point. But anyway, we gave the talk. And we had, one of the things we had done was we had used formaldehyde to stabilize the denatured component of the DNA. And what we had discovered was that we could partially denature DNA and maintain it partially denatured. And the thing about that was that in those days, we were so primitive that we did not know, when you started to denature the DNA molecule, whether it came apart at the ends and unwound, or whether it really came apart where there were the fewest hydrogen bonds keeping the 2 strands together. And we had a faculty member down the hall, Mike Beer, take electron micrographs of this partially denatured DNA. And what we would see, and this was with negative staining, you'd see a little strand of DNA, then you see a puddle, see a strand coming out the other end, another puddle, etc. And so it was pretty clear that the DNA was denaturing along its length, most likely in the AT-rich regions. And I presented that in my talk. And at the phage meetings in those days, guys used to stand up, say, that's a lot of nonsense, or whatever they would say, usually not so kind. Or they'd say good things. Anyway, Max Delbruck jumped up, and he said, would you mind going over that once again for me. So I went through it again. And Max got this great grin on his face. Said, I like it. And he sat down. And I thought, well, we're not going to ever do any better than this, and I should have quit probably right then. But anyway, so it was a very heady experience, and then I started regular medical school, which was not that kind of experience. It was kind of old-fashioned. The genes were described as complex proteins in the nucleus. And so I got through the first year, and I became a graduate student, and I went through graduate school, and then I decided there had to be more in medicine than I had seen, so I went back and I finished medical school and did an internship in pediatrics, which was terrific. And I went to the NIH. At the NIH, I first worked with Arthur Weisbach for 1 year, and then he was lured away by the Roche Institute, and so I switched over to the laboratory of biology of viruses with Norman Salzman, with whom we'd already been collaborating because we had started working on vaccinia DNA polymerase. Anyway, the point of this part of the story is that one night there was a party in the lab, and another member of the lab, and this was a schizophrenic lab with two factions, but Jim Rose, who was in the other faction of the laboratory, and I started talking at this party, and I don't know what we were drinking, but anyway, we had quite a convivial conversation, and I told him a joke which he loved. And the next morning, he came up to me very agitated, and it turned out he and Aaron Shatkin had just published a paper characterizing the purified DNA of adeno-associated virus, a small parvovirus. And it was linear duplex. And they finished their paper by saying, since it's a linear duplex, when it's purified, that likely represents the state of the DNA in the viral particle. And suddenly, this crazy guy, Lionel Crawford in London, had published a paper. He said, there's something wrong with this conclusion, because if you look at adeno-associated virus, it's the same size as viruses that contain only a linear single-stranded DNA of half the molecular weight of the DNA that had been characterized. So I said, maybe, as crazy as this sounds, there are particles that contain the plus strand and other particles that contain the minus strand. And Jim was very distraught, and he said, what can we do to disprove this notion? And I said, well, let me think about it. And the next day, after a good shower and being able to think about how to solve this problem, I came back and I said, well, if we can make density labeled AAV DNA, then we can do it. And what we did was the Mendelssohn-Stahl experiment. So we took heavy particles and light DNA particles, and if we purified the DNA separately and put them in a cesium chloride gradient, we got heavy DNA and light DNA. And if we mixed the particles together and then purified the DNA, then of course we got DNA of hybrid density. And so that was an easy experiment to do, and I didn't expect that would lead to 45 years of working on this little virus. So the question is, who's had adeno-associated virus? Who's been affected by it? One guy knows, and he doesn't count. Anyway, it turns out we've all had it. Now, why don't we know? We don't know because it doesn't cause any disease, as far as we know. And the only reason for a long time a lot of people study viruses is because they cause disease. But I thought it would be a nice virus to work with because it was small, and you might really be able to dissect it and figure out everything. Small genome of about 4,500 base pairs or bases. But the problem is, of course, that was a very naive notion, because a small virus And a large virus like herpes or pox viruses have to accomplish exactly the same things. So if you can accomplish everything that a herpes virus can with a genome of 4.5 kb, you're very devious and tricky. And so it's been an interesting career in terms of trying to dissect this out. Now, remember, this virus doesn't cause any disease. Fortunately, the NIH was fairly generous over the years, and even NSF and the Cancer Society, for reasons I'm not totally clear on, also supported us. Of course, some of my proteges and colleagues decided that it actually could be used as a vector for gene therapy. And everybody else laughed. I mean, AAV was because of the lifestyle of this virus, in that it requires a helper virus in order to go through productive infection. It was also called almost a virus. And everybody, you know, I think when we started talking about it as a vector, they all, adenovirus was terrific, and every other virus you could think about, retroviruses. And so they say, oh yes, if they remembered, we can also do AAV. And of course, what the situation we've gotten to now is the fact that I would say it's certainly in the top 2 in terms of viral vectors. And in the hands of people at Florida and some collaborators in Philadelphia, it's actually been used to cure blindness. And several other labs have been able to do that at the same time. And it's been used to cure hemophilia B. In fact, it actually has been approved in Europe as a standard therapeutic for a particular disease, which I suspect is head and neck cancer. But I think that I don't know of even a better example of what happens with basic research. All the work that we did was because we're interested in the biology, without any notion that suddenly it would become a therapeutic agent. And so I think there's a very good story there in that respect. Anyhow, when I was at the NIH, I was offered the chance to go back to Hopkins on the faculty in microbiology by my patron there, Barry Wood. Barry was, everybody know who Barry Wood was? Nobody remembers. Anyway, Barry was Harvard's last All-American quarterback. And he went to Hopkins to medical school, and 10 years out of medical school, he was the chair of medicine at WashU. I claim was the reason that WashU may have the best medical school in the country today. He's got everything started. And then he came back to Hopkins as the VP. As he told me, he said he had no idea what the hell he was doing for 5 years, and finally they let him be the chair of microbiology when he can go home at night and think about his experiments. And I think that parallels some of the title, maybe, of this talk. So I was at Hopkins for 6 years on the faculty. I was sitting between Ham Smith and Dan Nathan, so that was my laboratory position. And of course, all the time they were doing experiments that won them the Nobel Prize in '78 on recombinant DNA. In 1975, I had been a Hughes investigator, and Barry died, and things changed, and so the message was given that since your patron is no longer the Hughes person here, you ought to look for other support. And that was the year that the NIH decided not to do RCDAs anymore. And so I applied to the Cancer Society, and they sent a man named Harris Bush, who was the chair of pharmacology at Baylor, a notable character. And Harris came, and he was very convivial, nice, and he said, "What the hell are you doing here?" And I said, well, you know, I said, "What do you mean, what am I doing here? That's why you had the visit." But anyway, he said, no, no. He said, you know, you're an MD, PhD. Why aren't you a chairman? And I gave him an even more amazed look, and he said, well, you know, you know, you haven't been asked yet, obviously. He said, but you will be. He said, you know, at your age, I was offered the job at Baylor. He had been at Yale as an associate professor, and he went. And so, in fact, a year later, I was offered a chair at Florida. And the problem was that I used to tell my chairman, it was Dan Nathans by that point, what to do all the time. And much of the time, he actually listened and did it. So it was really an incredible situation. But I knew that if I didn't accept a position to assume the responsibility for all my great ideas, that I ought to shut up and quit telling them to people, you know, and not being willing to assume the responsibility. So I left Hopkins and I went to Florida, and I think the message, and let me just say, the notion was, can you create an environment which is really conducive for the development of young people, students and young faculty? And that was really, it was an interesting challenge from my point of view. I mean, when you're younger, you don't realize what the real problems might be. And so I tried to do that. And the original guys most of them are still there in senior positions, and they've been very, very successful. 2 of them are eminent scholars, and 1 chairs the department. And before I left Hopkins, this is for the Rosalind Franklin Society, there was a young woman who was working as a fellow in our department, and she needed to find a different position. And it turned out I had been riding the bus during the gas shortage with a fellow who was in neurology, Bill Narayan, and I said, Bill, you ought to take this young woman on in neurology, because he was looking for a molecular biologist. And so he did that, and this woman's name was Janice Clemens. She became an extremely successful retrovirologist, and today she's the Dean for Faculty Affairs at Hopkins. And that was one of my more successful matchmaking efforts, I would say. Anyway, I was at Florida. When I got to Florida to be the chair, the dean said, "This is a job you want to take for 6 to 10 years. It takes 6 years to get anything done. At 10 years, it's time for fresh blood." So I said, "Yes, sir." It seemed like a long time to me. But at 8 years, well, let's say in the 7th year, I actually got a sabbatical. And I went to Israel to the Weizmann Institute, worked with Ernest Winokur, one of the great SV40 virologists. Maxine Singer and Dan Nathans had both learned how to get into SV40 work in his lab. And the consequence of my being there was that he spent the rest of his career working on AAV, so I felt very subversive. And the reason was that after I'd been there about a month, a woman who was about my age, actually, but was a graduate student, came up to me and said, "Your project is going to be my project." I said, I didn't want to argue. What do I know? And so she started working with me as a woman named Zahava Grossman, and so Ernest actually had a look at her thesis in some detail, and he got intellectually stimulated by what he read. And so Zahava and I are still friends. We had dinner not so long ago. She's now retired. She was the head of HIV diagnostic lab for Israel, and really had a lot of great epidemiologic data from that. Anyway, I came back to Florida, and now it was 8 years, and I was offered the position of chair at Cornell. And I thought, well, I'll get to be at an Ivy League school. I'll get to live in a world-class city for some fragment of my life, because there aren't too many in the US, in my opinion. And so I went up to Cornell, And that was a great experience. I had a very good time. I went up there. There were 2 faculty in the department, and only one of them was a microbiologist. So it was a chance of starting from scratch. And most of the people that I recruited in the beginning are still there. One of them deserted to Rockefeller, is a member of the National Academy these days. But all of them really did very well. And we formed a unified graduate program with Sloan Kettering, which was kind of interesting and worked very well. So that was a very good experience. But all of this time, I had been involved in extracurricular activities. And one of the things I was very active in was the AAMC, the Association of Medical Colleges. And eventually I got to be the chair of the Council of Academic Societies. And then my patron there, Bob Petersdorf, who was the president, arranged for me to become the chair of the association. And so I had to start thinking about, the things deans think about and all this stuff, and public health policy. And it's, after I saw all the deans, when I was growing up as a young faculty member, forgive me if I offend anybody here, the definition of an assistant dean was a mouse studying to be a rat. And so the notion of being a dean was not particularly, I don't want to offend anybody, but it was questionable. It was questionable. I think, in fact, since I had actually been in the dean's office at Hopkins, et cetera, that actually I didn't feel that badly about it. But it seemed to me, again, it was a chance of asking whether at a medical school level you could create an academic environment that would promote the development of young people in an academic sense. And so I was offered the job at Florida, and I went back down to Florida. And for a variety of reasons, within a year, I also was the VP for Health Affairs. And it was actually fun to be the dean because it was academic, in addition to all this stuff worrying about taking care of the group practice and that sort of thing. But dealing with the hospital as the vice president was more challenging and less gratifying. And I should have learned from that, but I didn't. So after about 5 or 6 years of doing that, I was offered the position at Mount Sinai, which I would say was in less than good condition at that moment, since the hospital lost $75 million the year before. And so I went up to try that out, and that was not a great experience from my point of view. And so while I had been the dean, when I had started as the dean, we were in just starting a capital campaign, and the College of Medicine reached its goal within a year, and we still had 3 years to go. So the president, who was a rambunctious guy named John Lombardi, said to me, well, now your goal is 50% higher. And of course, we were happy to get all the money we could get, so having a better goal was even better. And he said, but you need a new project to justify that. So he said, you keep telling me how important genetics is, so we'll have a genetics institute. So, and it'll have a building eventually. And so after my experience at Mount Sinai was less than great from my point of view, the university invited me back to be the head of the Genetics Institute. Now, I don't have to tell you about the Genetics Institute because Dr. Schass described it perfectly. This morning, we built a building. We've got about 40 faculty who are genetics types. The notion of genetics, the requirement to become a member is to say you're interested in genetics for whatever reason. And so we had people even we had ethicists, we have physicists, we have statisticians, engineers. and all the usual suspects who are members. There are about 250 faculty in Florida who are members of the Institute. So I got to go back, supervise the construction of this building, which was the largest research facility on the campus and probably in the state, and recruit new faculty, and it's been very, it was very satisfying. I stepped down after 9 years, remembering the 6 to 10 year admonition that I got when I went there the first time, and they went out and have recruited a new person who's scheduled to arrive in a week or so. So that was actually pretty much it. I think the issue is that, you know, so you have a building, you build or 2 buildings. But it's the people that are critical, that really make the place. And so the people that you've been involved with, I think, are really nice. One of the, when I was the dean, I really thought that what we were lacking, one of the things we were lacking was Xray crystallography capability. And so it turned out I knew a couple who were in England who were young, and they're both Xray crystallographers, and they, one of them at least worked on parvoviruses, coincidentally. But anyway, we brought these people, and they set up Xray Crystallographic Center, very successful. I guess I would now describe the woman involved as an African American. I don't know, she was an African Anglo, I guess, at that time. Had grown up in the UK and back and forth in Nigeria. Anyway, this woman is sufficiently successful that at the moment she's on the advisory council in IAID and is a real power. And it's just one of the great success stories, and that's the kind of thing that, from my point of view, having been an administrator, I probably take the most satisfaction from. As far as the research goes, what's happened with AAV is very satisfying, but I'm going to finish with one quick story here. The first day I was at Cornell, one of the faculty came to me and said, one of our students is taking an oral exam today, would you like to sit in on it? Graduate student. I said, sure. So I sat down in this exam, and this young woman got all these questions from 4 or 5 members of her panel, and she answered them. She was really quite polished. And at the end of the exam, they said, well, Dr. Berns, would you like to ask any questions? And I said, I think I would, actually. And I said to her, what department are you in? She said, immunology. I said, is that the name of the department? She said, oh, no, it's microbiology. I said, well, you answered all these questions, but you didn't get a single question that have anything to do with microbiology, so I thought maybe I would ask you a microbiology question. She said, okay. So I said, do you ever go next door to the Rockefeller University? She said, sure, I go over there all the time, a couple times a week. I said, well, do you know who the president of the Rockefeller University is? No idea. So I said, well, his name is Joshua Lutterberg. Do you have any idea why he's the president of the Rockefeller University? No idea. So I said, well, can you tell me anything about bacterial conjugation and phage transduction? And she gave, you know, I mean, she knew the material. Gave me a perfectly good answer. I said, well, just to increase your learning experience, you should know that he discovered both of those phenomena and received the Nobel Prize when he was 37 years old. So my guess is she still remembers Joshua Lutterberg. And what I remember is that at that moment I gave up any concern about ever becoming famous. Just a false wish to have, as far as I could see. So thank you all very much. Told you everything you need to know. Oh, yeah.
Really serious question.
Yes.
I want to know which newspaper you carried in Cleveland and when that was approximately, because our middle son, named Neil, was a newspaper carrier in Cleveland. It was the Cleveland Press, actually, not the plain dealer, in the snow, and he lives in Florida, so there must be some.
Well, so you should know that I started after 4th grade delivering the Cleveland Press. Okay. And that was the afternoon paper. And it was a paper that carried more lines of daily advertising than any other paper in the country at that time. It had a great editor named Louis Seltzer. And then a couple of years later, I switched over to the plain dealer because I could deliver it before I went to school and I didn't take up my afternoons. And the epitome was when my route was in a big complex of apartments, so I actually could stay inside for most of the time, and that was much better. Yes?
One thing that really strikes me is your treating experience, like many of the scientists nowadays who are doing very medical and related research, is on very basic phage biology, and that's what...
My scientific, yeah. I would say, though, that going to medical school and the curriculum that we had then at Hopkins, and especially being a pediatrics intern, in what's known as was known as Harriet Lane, you got a lot of medical, experience, and it's made a difference from the way I look at things. From my point of view. So I think from what I did eventually and the kinds of jobs that I took, I think that was very much colored by having finished medical school.
So then what's your retroactively, how much do you think the basic research has contributed there?
Well, so I told you that when I started on the faculty at Hopkins, Ham Smith was on one side and Dan Nathans was on the other side, okay? 2 guys who are board eligible in internal medicine, no PhD training. Smith did 6 years of being a fellow with Mike Levine at Michigan in human genetics. That was doing phage studies, so he had the same experience I did probably. Dan did a postdoc, and I'm going to block on the fellow's name, but it was a Nobel Laureate at Harvard. And there are times because when Dan was doing his key experiments, we would talk a lot, and sometimes I would suggest things to him as a good control to really, and which he did. And I mean, that's the way he was. He just picked up. You don't have to get a PhD to win the Nobel Prize in medicine. That's my conclusion from all of that experience. For a guy like me, I think it was really helpful to go through the discipline of taking the courses. I think for other people, Nathans and Smith, they were well able to do it on their own.